take a look at notes from students who had my class. So we are at week 3 and 4. So we talked about membrane transport and intracellular trafficking. We talked about transporters, channels, and some of these are activated by ionotropic receptors. And we're going to talk a little bit today about mechanisms of how these receptors activate vesicle fusion and endocytosis. We went through these vesicles and endocytosis. Give me a second, I need to get us. I moved to a very cold place. So we talked about how vesicles, which carry proteins, carry these proteins and fuse with the plasma membrane. And then once they fuse with them, they remove whatever materials came from the Golgi by the process of exocytosis. So we're going to start how receptors are important in directing this type of reaction. We're also going to talk receptors are also involved in moving materials across transporters and channels. I hope Mike picked up 2 handouts, so you can get handouts from Mike, which give a few notes on some of the things that we've talked about previously. It does not mean which has been put in the course outline from which these two are derived. So we're going to look at the concept of receptor and signal transduction, particularly how this happens in the synapse as an example. Receptors are in many, many tissues. And when a ligand binds a receptor, it induces a number of intracellular pathways. So please don't think that receptors only assist in synaptic transmission. By now, of course, I hope you know what the synapse is. I'm not going to go into detail. I'm assuming you did that in physiology. And in the synapse, the neurotransmitter is the one that is responsible for action, and this is by reversing the potential. Remember, we said the membrane potential is at the resting phase until it is activated by neurotransmitter, and how these are integrated so that you get responses. Obviously, neurotransmitters are found between 2 neurons, the axons of the 2 neurons, and also you find them in other places like glands, so that they bring information that a particular hormone needs to be released by vesicle fusion with the plasma membrane, or a particular hormone, or in the junction between the nerve and skeletal or smooth muscle cells. So these are very important, and I'll use this as an example to give for understanding the concept of receptors and signal transduction. So this will be today and tomorrow's lectures. So we're going to look at how ligand-gated channels, they open to a chemical signal, and this chemical signal is what is referred to as a ligand, instead of changes to the membrane potential. So ligand-gated channels, ion channels, are responsive to ligand binding to the extracellular receptor. And the most important of these ligand-gated ion channels in the nervous system is the class which is activated by binding of neurotransmitter, transmission of neural signals. That is what the word neurotransmitter means. But these neurotransmitters are chemicals. So you need to be able to identify the difference between voltage-gated channels and ligand-gated channels. In voltage-gated channels, we look at the sodium channel, we looked at the calcium channel, we looked at the potassium channel, and we looked at the chloride channel. And here, in the potassium channel, you have a sensor which senses the voltage on the membrane. We have a sensor on the sodium channel, we have a sensor on the calcium channel, and we have a sensor on the chloride channel. So these are voltage-gated channels. When the voltage changes either on the outside of the extracellular part of the lipid bilayer or the plasma membrane, or in the intracellular part. We went through all those details. Now, when it comes to ligand-gated channels, I think we talked about this. We talked about a receptor on the extracellular side of the integral membrane protein, which is known as a receptor. So in this particular example, glutamate is the chemical which binds this receptor, and when it binds this receptor, it causes opening of this channel, and when the channel is open, sodium ions go out and potassium ion comes in. This is the neurotransmitter receptor, activated by glutamate or acetylcholine or any other neurotransmitter. This example is glutamate only. Then you have calcium activated potassium channels, and these calcium activated channels are activated intracellularly. Calcium is released, binds the inside part of the integral membrane protein, which is the receptor, and this calcium binding will cause this channel to open and potassium ions to be exuded from the cytoplasm into the extracellular space. Then you have cyclic nucleotide. Cyclic nucleotide are cyclic AMP, cyclic GMP. All those are cyclic nucleotides. It will be activated by a cyclic nucleotide, again, on the intracellular side of the membrane, and this causes the channel to open and cause movement of sodium into the cell and potassium out of the cell. These molecules, the cyclic gated channels, are activated by cross receptor or by other signals coming from inside the cell. So you can see the difference between neurotransmitter receptors and calcium activated potassium channel and cyclic nucleotide gated channel. So it's important, number one, to know the difference, the voltage gated channels and the ligand gated channels. In the ligand gated channels, there are a number of mechanisms. In the voltage gated channels, you can see the voltage is controlled by the charge of this, and we went through the electrochemical balance in the extracellular and intracellular side. So now we are moving on to the next group. I put this up so that you don't get confused between the two. So these channels, they are essential for synaptic transmission and also other forms of cell-to-cell signaling phenomena. I think you had in cell biology, when one cell signals to the other, what is that mechanism called? Can somebody type it in? When you get a signal from one cell and it signals the adjacent cell or another cell within the same tissue, what is it called? Anybody? Can you hear me? The silence is deafening. No? Signal transduction is also a generic. Specifically, I asked, when a signal moves from one cell to an adjacent cell or another cell within the tissue, what is endocrine signaling? Endocrine. Because there's paracrine, there's endocrine. Autocrine? Brian, what's the difference between autocrine and paracrine? Valerie, can you tell Odhiambo Brian the difference between autocrine and paracrine? Valerie, are you there? Chalo, Daniel? Daniel? Okay, Valerie, go ahead.
Endocrine?
No, autocrine. I'm saying autocrine and paracrine. What's the difference? Because Odhiambo seems to think it's autocrine. No? No. Yeah, we know that cell-to-cell signaling is also called intercellular signaling, right? But it is specifically referred to as paracrine as Valerie said. It is paracrine signaling. So, where does autocrine signaling occur? That's my question. Where does autocrine signaling occur?
Hello, it occurs on distant cells.
Autocrine?
Yeah, occurs on distant cells, while paracrine has nearby cells.
Paracrine is nearby cells, so if autocrine is distant cells, what is endocrine? What is endocrine? What is endocrine? Valerie. Okay. So, I've typed it in there. Autocrine, as in automatic, is releases a signal, and the signal acts on the same cell that released it, not a distant cell. Distant cells is endocrine. Adjacent cells or cells within the same tissue is paracrine. So this is stuff that you did foundationally, and all of my cell biology class, I should go back and fail you, because that was the whole point, so we don't have to do this song and dance. Anyway, where ion channels underlying the action potential will typically allow only one type of ion to permeate. Channels which have been activated cellular ligand come from inside the cell and cyclic nucleotides. So these, the extracellular ligands, are usually less selective, and these ones will allow two or more types. If you look at this here, this is intracellular, and this is intracellular chemical signal. Here it is activated by So ligand-gated ion channels include those that have been activated by the extracellular presence of neurotransmitters. An example of a neurotransmitter such as cyclic adenosine monophosphate or cyclic guanine monophosphate, cyclic GMP or cyclic AMP. Now, what are intracellular second messengers? Intracellular second messengers. OMA. OMA, Trevor Grantlin. OMA, are you there? Anybody who can help OMA? Anybody who's ready to take a wing at it? I think Valerie is very brave. Because sometimes when you make a small mistake in answering a question, you're able to correct something which you have been erroneously carrying along. Wanjiko Christabel Nyambura, are you there? If you're not responding, I'm marking you as absent. Wangeshi Ndirangu, if you're not responding, I shall be marking you as an absentee. So Wanjiko can, with second messengers such as calcium and the cyclic nucleotide AMP or GMP. Such channels can be selective for specific ions such as potassium or chloride, or they can be permeable to all physiological cations. Okay? So they are not as stringently specific as the others we've looked at. The main function of these channels that respond to second messengers, is to convert the intracellular chemical signal into electrical information. And this is particularly important in sensory transduction. Sensory transduction is in many organs of the body: the eye, the ear, the nose. So where channels gated by cyclic nucleotides convert odors, that is smell, light in the eye, into electric signal. So I'm going to give you your 2-paragraph assignment. Paragraph 1. Explain how paragraph 2. Explain how light signals activate receptors in the eye. A. What are the receptors for light in the eye? And B. What are the molecules that are activated following ligand binding of the receptor. This is going to be interesting because in the eye, you have receptors, and these receptors are for light. Okay? In the nose, sorry, this is typo, it's not explain, it's explain how in the nose it is smell. And how are these receptors for smell? That you can smell, and that is very good pilau, that's a very good curry. Oh no, that smells awful. So, how are these receptors activated? And I don't want you to go and photocopy and plagiarize all manner of things. I want you to explain it to me. And you've seen how in your exam, those of you who go and photocopy these things, you're completely clueless when it comes to answering the same question in the exam setting because you just went and photocopied. So you need to understand. You also need to explain to me how light signals will activate receptors in the eye. And in doing so, I would like to see what are the receptors for light in the eye. And once those receptors have bound the ligand, I'm not going to tell you what the ligand is because light is not a chemical. What are the molecules that are activated intracellularly following binding of the receptor? Okay? So those are two questions. This assignment should be in my office by Wednesday next week. This assignment should be in my office by Wednesday next week because we're going to be doing a lot more detail when we go to pain or Nochi receptors. I need you to do this. So this, as I said, this process of the channels is to convert intracellular signal chemicals into electrical information. So some intracellularly activated ion channels are in the surface of the membrane. They are not in the intracellular side. But there are others, like the calcium I showed you, which are intracellular membranes, such as the endoplasmic reticulum. So these latter channels are selectively permeable to calcium, and they will regulate the release of calcium from the lumen of endoplasmic reticulum into the cytoplasm. The calcium which is released can then trigger a spectrum of cellular responses. Why? Because this calcium which has been released this way is a messenger molecule in its own right. No, you cannot submit to my email hard copy because I need to print all these things, and sometimes you people double submit. That's not going to happen. So I would prefer this is okay. So what are ionotropic receptors? They are also referred neurotransmitter gated or ligand gated channels. They are ion channels that responds to the binding of a neurotransmitter or a chemical ligand. They are primarily located along. I hope you know what dendrites and axons are, but they can be present anywhere along the neuron if there is a synapse. Why? Because this synapse has the presynaptic knob, it has the synaptic cleft, which is a space, and then it has a post-synaptic cell. So to cross that chasm between the pre and post is where you have this synapse. And that is important because you need these neurotransmitter to be released by the pre and bind the receptors on the post. So ligand-gated channels are important for receiving incoming information from other neurons and in the neuromuscular junction from a neuron to a muscle cell. I'll let you copy that intro on what ionotropic receptors are because I'm not going to keep coming back to define what they are. So I'll give you a the ones indicated, the spikes. This is the cell body. That's a nerve. Those are the myelin sheaths. And the signal goes from one place to the other until it reaches the terminal nuclei, and the signal then moves into that, or the electrical impulse then moves to the target. Just in case you missed your anatomy class, although ionotropic receptors are ion channels, they open in a different way than the voltage-gated ion channels, and I've gone through that, which is needed for the propagation of an action potential. The action potential is an electrical impulse or a nerve impulse. The ionotropic receptors are ligand-gated, which I've shown you, and this means that a specific molecule, such as a neurotransmitter or a ligand, must bind to the receptor to cause the channel to open, and once the channel opens, it will allow the flow of ions. Recall that voltage-gated channels open only in response to the membrane potential reaching threshold. The receptors can only be opened by a specific ligand. Neurotransmitters and receptors fit together like a lock and key, and which you saw in enzymology. Only certain neurotransmitters are able to bind to and open certain receptors, and we'll be going into a lot of detail into which neurotransmitters bind to which receptors, and then when the receptors are open, what is the signal that is transduced from them. These receptors are often composed of more than one subunit. The ligand or the signal will bind to one or more subunits, resulting in a conformational change. And I suspect you talked about conformational change. The molecule changes in its physical arrangement. And this conformational change, it will literally turn around, is what opens the ion channels. And as a result of this, ions will rush in or they will rush out of the cell down their concentration gradient. So we've combined signals, binding receptors. These receptors have more than one subunit, so this is an integral membrane protein that crosses the lipid bilayer several times. And once these receptors bind either to one, depending on the signal, or more, you get a change in the protein orientation. And this change in protein orientation will result in opening of the ion channels, and one cellular side and a part on the intracellular side. When the neurotransmitter binds to specific sites in a lock and key hypothesis type method, the channel will open, and when the channel opens, it will allow the ions to rush through. Okay? This neurotransmitter has been released in the cytoplasmic or synaptic cleft by the cell which is here. So here imagine there's this exact thing, but from this side The neurotransmitter was released by vesicles which bound the membrane integrated with the vesicle, and then you had exocytosis. Okay? Then you had exocytosis of the neurotransmitter. It crosses the synaptic cleft to the membrane of the postsynaptic cell, and there it will find its very special and own receptor, and when it binds, it will allow ions to pass through, and by allowing the ions to pass through, it will cause a change in the electric potential, and then will be propagated from one presynaptic cell to the postsynaptic cell. Features of postsynaptic receptors have several general receptors. Postsynaptic new receptors may be directly or indirectly coupled to channels. The example that I showed you is a direct receptor because it is right on the channel itself. This is directly coupled receptor. So directly coupled receptors are called ionophores, and these are ionotropic receptors, and they produce very fast postsynaptic responses. Then you have the indirectly coupled receptors, and these are indirectly because there is a second messenger which is released following binding of the receptor with the ligand, and they are called the metabotropic receptors. And this is in the category of G protein coupled receptors, and these produce slow postsynaptic responses. Why? Because of the indirect mechanism. It has to go through a messenger. It's like if I tell somebody to come and give you this lecture, it's going to be a little bit more difficult because it's going through a messenger and not directly from my own mouth. So it will take time for that person to actually be able to convey the message that I intended to convey. So it's better to hear it from the source. So metabotropic receptors go through G protein coupled receptors, and they produce slow postsynaptic responses. And this G protein is the G cyclic GMP or the cyclic AMP that I talked to you earlier about. Have we got this concept? Can I have a yes or no? Or a query? Feel free to interrupt if you need to query. Live. Remember my mantra. When you're in school, there's no such thing as a stupid question. It is a specific sites on the receptor. Okay? So this integral membrane is the receptor. Then a whole bunch of signals which I'm not going to talk to you about go through the receptor. This will do probably in masters. You see all these steps. It's not just simple, direct, but it goes through some steps. It will activate G protein, and then this G protein binds to an effector protein, and these effector proteins require ATP or energy, and once the effector, which actually effects the signal that came in, will release second messengers, which will activate enzymes, and these will come and open an adjacent receptor, and once they open open a channel, and once the channel is opened, the ions will flow through. Look at the difference. This receptor itself is not opening. It is causing the activation of a G protein. That G protein will then activate an effector. No, G proteins are not enzymes. G proteins are not enzymes. G proteins are cyclic guanosine monophosphate. Okay? So, the G protein here is very different from these little enzymes. You see the difference? So the G protein, it sits bound to this receptor. It sits over here so that this effector does not float around being exposed to ATP and activating enzymes. It has to be bound in this conformation. It is only when the signal, the neurotransmitter, binds the extracellular side or binds to the specific site that the signal comes to this G protein, and when it activates this G protein, the effector which is bound sort of slides, undergoes a conformational change, and when it undergoes that conformational change, it exposes sites, maybe in here, which will allow ATP to bind, and once that ATP binds, you get activation of the second messengers, which could be in the type of enzymes or other and bind